Inhibition of IL-11 signaling extends mammalian healthspan and lifespan. Inhibition of IL-11 signaling extends mammalian healthspan and lifespan. For healthspan and lifespan, certain pathways like ERK, AMPK, and mTORC1 are crucial, and inflammation is a central hallmark of aging. This study examines whether IL-11, a pro-inflammatory cytokine of the IL-6 family, negatively impacts age-related diseases and lifespan. As mice age, IL-11 is upregulated in various cells and tissues, affecting the ERK, AMPK, and mTORC1 axis and contributing to aging pathologies. Deletion of IL-11 or its receptor protects against metabolic decline, multimorbidity, and frailty in older mice. Administering anti-IL-11 to 75-week-old mice for 25 weeks improves metabolism, muscle function, and reduces aging biomarkers and frailty. In lifespan studies, genetic deletion of IL-11 extends mice lives by about 24.9%. Treatment with anti-IL-11 from 75 weeks of age until death extends the median lifespan of male mice by 22.5% and female mice by 25%. These findings suggest that anti-IL-11 therapy could be used to address aging pathologies in humans. The major signaling mechanisms regulating lifespan across species include ERK, STK11, also known as LKB1, AMPK, mTORC1, and IGF-1 insulin modules. These pathways are disrupted in old age, activating hallmarks of aging like mitochondrial dysfunction, inflammation, and cellular senescence. The AMPK-mTORC1 axis is particularly important for metabolic health in aged organisms, especially in adipose tissue. Therapeutic inhibition of mTOR extends lifespan in mice. Most aging studies have focused on lifespan extension, particularly in simpler organisms like yeast, worms, and fruit flies. However, lifespan extension does not always mean a longer health span. Integrated studies on both healthspan and lifespan are needed. Laboratory mice are suitable for such experiments because they show aging pathologies similar to humans, and lifespan studies are well established in mice. Chronic sterile inflammation is increasingly recognized as important for aging pathologies and is a central hallmark of aging. Aging is associated with a dysfunctional adaptive immune system characterized by immunosenescence and thymic involution, along with inappropriate activation of innate immune genes like IL-6. Pro-inflammatory signaling factors, such as NF-kappa B and JAK-STAT3, are specifically implicated in aging, and JAK inhibitors can alleviate age-related dysfunction. We proposed that IL-11, a pro-inflammatory and pro-fibrotic member of the IL-6 family, may promote age-related pathologies and reduce lifespan. This idea was based on studies showing that IL-11 can activate ERK-mTORC1 and/or JAK-STAT. The observation that IL-11 is upregulated in older people, and the fact that IL-11 is increasingly recognized for its role in senescence, a hallmark of aging. Using various genetic and pharmacological approaches, we tested the hypothesis that IL-11 signaling negatively affects healthspan and lifespan in mice. IL-11 is upregulated with age. We measured IL-11 expression in the liver, visceral gonadal white, adipose tissue, and skeletal muscle in mice over a time course of aging. This revealed progressive upregulation of IL-11 in all tissues. With age, there was progressive activation of ERKP90-RSK, inactivation of LKB1-AMPK, and mTORP70S6K activation, forming an IL-11 signaling module in liver and muscle. IL-11 upregulation was confirmed in livers, visceral gonadal white adipose tissue, and skeletal muscle of 2-year-old male and female mice. To identify the cell types expressing IL-11 in old mice, we examined the tabula maris senes dataset, which was uninformative, likely due to low expression levels of IL-11, as it is largely translationally regulated. We performed immunohistochemistry in tissues from aged IL-11 EGFP reporter mice. In 2-year-old IL-11 EGFP mice, IL-11 was found in parenchymal cells, such as hepatocytes in liver, adipocytes in visceral gonadal white, adipose tissue, and myocytes in skeletal muscle. It was also seen in stromal, epithelial, and endothelial cells across tissues and in nerves and skeletal muscle. Thus, IL-11 is expressed in various cell types in different tissues with aging. IL-11 is associated with senescence. We further explored the relationship between IL-11 upregulation and ERK-mTOR activation in tissues of old mice by studying 10-week-old and 2-year-old IL-11 receptor knockout mice and wild-type littermate controls. Immunoblots of liver, visceral gonadal white adipose tissue, and gastrocnemius extracts from old wild-type mice showed increased phosphorylated ERK, P90RSK, and PLKB1, leading to reduced PAMPK, and increased levels of PMTOR, P70S6K, and PS6R. Levels of canonical senescence markers P16 and P21 were also increased. In contrast, these phosphorylation levels and senescence markers were similar between old IL-11 receptor knockout mice and young wild-type mice. Deletion of IL-11 receptor improves metabolism, compared with wild-type littermate controls. 2-year-old IL-11 receptor knockout mice had lower body weights. Female IL-11 receptor knockout mice had decreased fat mass and increased lean mass. Both male and female IL-11 receptor knockout mice had slightly higher core body temperatures than wild-type controls. Old IL-11 receptor knockout mice had lower indexed visceral gonadal white adipose tissue mass and increased indexed gastrocnemius mass. Liver indices were similar between genotypes, but liver triglyceride levels were lower in IL-11 receptor knockout mice. Serum cholesterol and triglyceride levels were higher in old wild-type mice than in IL-11 receptor knockout mice. Livers of old IL-11 receptor knockout mice of both sexes showed reduced expression of pro-inflammatory and fatty acid synthesis genes, serum alanine transaminase and aspartate aminotransferase levels. Markers of hepatocyte damage were increased in old wild-type mice, but not in old IL-11 receptor knockout mice. Telomere lengths and mitochondrial DNA copy numbers, biomarkers associated with biological age, were preserved in tissues of old IL-11 receptor knockout mice. IL-11 induces senescence in human cells. IL-11 is linked with senescence, so we explored its direct effects on senescence in human cell types. Stimulation of human fibroblasts or hepatocytes with IL-11 activated ERK-mTOR, increased levels of P16 and P21, and reduced expression of PCNA and cyclin D1, which was prevented by U126 or rapamycin. Supernatants of IL-11 stimulated fibroblasts had increased amounts of senescence-associated secretory phenotype factors, which were inhibited by U126 or rapamycin. Deeper profiling of mTORC1-dependent senescence-associated secretory phenotype factors in hepatocytes revealed significant IL-11-stimulated ERK and mTORC1-dependent regulation of many proteins. Accumulation of senescent cells contributes to aging pathologies. We modeled replicative senescence by serially passaging human fibroblasts with a neutralizing IL-11 receptor antibody or an immunoglobulin G control. Passage-dependent phosphorylation of ERK-mTORC1 DORF-V, NF, kappa B, and STAT3, along with increased senescence markers, were IL-11 dependent. Telomere lengths and mitochondrial DNA copy numbers were similar between early passage cells and late passage cells treated with the IL-11 receptor antibody, while these markers were reduced in immunoglobulin G-treated late passage cells. Basal metabolic respiration was impaired in immunoglobulin G-treated late passage fibroblasts, but remained similar to early passage cells in IL-11 receptor antibody-treated late passage cells. Deletion of IL-11 in female mice. To support the data from IL-11 receptor knockout mice on a mixed genetic background, and to further explore age-related effects, we studied young and aged female mice with deletion of IL-11 on a C57BL6J background. Immunoblots confirmed IL-11 upregulation across tissues in old age in this additional strain. Old female IL-11 knockout mice had lower body weights and fat mass and preserved lean mass. Their frailty score was lower than that of old wild-type mice, and their body temperatures were mildly increased. Lower frailty scores were driven by improvements in tremor, loss of fur color, gait disorders, and vestibular disturbance. Muscle strength was higher in both young and old IL-11 knockout mice compared with age-matched controls. Chronic inhibition of mTORC1 with rapamycin can cause glucose intolerance due to indirect inhibition of mTORC2. It was important to fully assess the effects of IL-11 inhibition on liver function, metabolism, and glucose utilization in old mice. Old wild-type mice had increased serum AST, ALT, cholesterol, and triglycerides, which were collectively mitigated in old IL-11 knockout mice. Glucose tolerance and insulin tolerance test profiles of old IL-11 knockout mice were similar to those of young wild-type mice. While old wild-type mice showed impairment, indexed skeletal muscle mass was greater in both young and old IL-11 knockout mice compared with age-matched wild-type mice. Old IL-11 knockout mice had reduced liver mass indices and liver triglyceride content. Indexed visceral gonadal white, adipose tissue, and subcutaneous white adipose tissue masses were reduced in old IL-11 knockout mice, while brown adipose tissue mass was unchanged. The expression of fatty acid synthesis genes in visceral gonadal white adipose tissue increased with age in old wild-type mice, but not in old IL-11 knockout mice. Similar to IL-11 receptor knockout mice, activation of the ERK-mTORC1 axis and upregulation of senescence markers were mitigated in visceral gonadal white adipose tissue and gastrocnemius of old IL-11 knockout mice. Pro-inflammatory gene expression was increased in visceral gonadal white adipose tissue of old wild-type mice, but not in old IL-11 knockout mice. Telomere lengths and mitochondrial DNA content in liver, skeletal muscle, and visceral gonadal white adipose tissue were reduced in old wild-type mice and attenuated by IL-11 deletion. Serum O6 levels were increased in old wild-type mice, but not in old IL-11 knockout mice. Deletion of IL-11 in male mice. Detrimental changes in body habitus. Weight, fat mass, lean mass, and frailty scores were mitigated, and body temperatures were mildly increased in old male IL-11 knockout mice. Muscle strength was higher in both young and old IL-11 knockout mice, compared with age-matched wild-type controls. Glucose tolerance and insulin tolerance test profiles of old IL-11 knockout mice were similar to those of young mice, while old wild-type controls showed impairment. Metabolic cage analysis revealed a higher respiratory exchange ratio in old IL-11 knockout mice compared with old wild-type mice. After starvation, refeeding increased the respiratory exchange ratio more in old IL-11 knockout mice, indicating better metabolic flexibility. Old IL-11 knockout mice were leaner, weighed less, consumed more food, and had similar levels of locomotor activity compared with wild-type controls. Calorie-losing enteropathy was excluded by bomb calorimetry. Sarcopenia was apparent in the muscle of old wild-type mice, but was less pronounced in old IL-11 knockout mice, which had greater indexed muscle mass even when young. Indexed liver weights were similar between genotypes. The most notable beneficial difference with IL-11 deletion in old male mice was reduced visceral gonadal white adipose tissue mass, while brown adipose tissue mass was similar across genotypes and ages. Enlarged seminal vesicles, an age-specific phenomenon in old male mice, were more common in wild-type mice compared with IL-11 knockout mice. Anti-IL-11 therapy in old male mice. Studies of germline genetic loss of function on healthspan and lifespan should be complemented by interventions in late life to identify phenotypes carried from younger animals and for translational relevance. In this study, some beneficial effects of IL-11 loss of function, such as increased muscle mass and strength, were apparent even in young IL-11 knockout mice. To achieve this goal, a neutralizing IL-11 antibody was administered to aged mice, and healthspan indices were studied. Mice receiving the antibody from 75 to 100 weeks of age progressively lost body weight, defined by reduced indexed fat mass. Impaired glucose metabolism was apparent across experimental groups at study start, but improved in mice receiving the antibody. Frailty scores were mildly increased at study initiation. But mice receiving the antibody did not show frailty progression, unlike untreated or immunoglobulin G-treated controls. Muscle strengths of antibody-treated mice at 100 weeks were higher than age-matched controls and higher than 75-week-old mice at the experiment's start. After 6 weeks of antibody administration, metabolic cage studies showed a higher respiratory exchange ratio in antibody-treated mice compared to immunoglobulin G-treated mice, indicating slowed age-associated metabolic inflexibility. Antibody administration was associated with higher core temperatures and increased food intake, while locomotor activity levels and fecal caloric densities were similar between groups. Mice left untreated or given immunoglobulin G had increased serum cholesterol, triglycerides, and IL-6, which were lowered by antibody therapy. Markers of liver damage. Hepatic triglyceride content and index liver mass increased in untreated and immunoglobulin G control mice, but these phenotypes improved or were reduced in antibody-treated mice. Indexed visceral gonadal white, adipose tissue, and liver mass were reduced, and indexed muscle mass increased in antibody-treated mice compared with age-matched controls and 75-week-old mice. Antibody-treated mice had diminished subcutaneous white adipose tissue and increased brown adipose tissue. Enlarged seminal vesicles were less common in antibody-treated mice. Fibrosis. A hallmark of aging and senescence was reversed in aged visceral gonadal white adipose tissue, skeletal muscle, and livers of antibody-treated mice. Visceral gonadal white adipose tissue of immunoglobulin G-treated mice had increased IL-11 TORC1 axis activation and higher senescence marker expression, while antibody-treated mice showed reduced ERK-mTOR activity and decreased P21 and P16 expression. Telomere attrition and reduced mitochondrial DNA copy number in untreated and immunoglobulin G-treated mice were not seen in antibody-treated mice. Anti-IL-11 therapy in old female mice. Anti-IL-11 therapy's effects on aging pathologies were also examined in old female mice. Female mice receiving the antibody lost body weight, while immunoglobulin G-treated mice gained weight. At the study's end, antibody-treated mice had lower fat mass, higher lean mass, and better glucose tolerance and insulin tolerance tests than at the start, while immunoglobulin G-treated mice showed the opposite. Frailty scores were similar between groups at the experiment start, but progressed in immunoglobulin G-treated mice, not in antibody-treated mice. Muscle strengths were greater than starting levels in antibody-treated female mice, and core body temperatures were mildly increased. Anti-IL-11 restores white adipose beiging. To further dissect molecular mechanisms, bulk RNA sequencing of visceral gonadal white adipose tissue, gastrocnemius, and liver from immunoglobulin G-treated or anti-IL-11 treated 100-week-old mice was performed. Mice receiving anti-IL-11 had significant gene set enrichment scores for oxidative phosphorylation and metabolism markers, while inflammation, EMT, and JAK, STAT3 signaling markers were reduced. In aged visceral gonadal white adipose tissue, genes associated with senescence were upregulated, an effect mitigated by anti-IL-11 therapy. This inhibition of senescence markers was also seen in muscle and liver tissues of antibody-treated mice. The gene most upregulated by anti-IL-11 in visceral gonadal white adipose tissue was UCP1, important for thermogenic beige adipocyte development in white adipose tissue. A larger beiging program was upregulated in visceral gonadal white adipose tissue of antibody-treated mice. Age-dependent upregulation of UCP1 and PGC1-alpha in male mice receiving anti-IL-11 was validated at the protein level. Age-related suppression of UCP1 expression in visceral gonadal white adipose tissue of female control mice was mitigated in female IL-11 knockout mice, and in mice of both sexes lacking the IL-11 receptor. A targeted assessment of mitochondrial gene expression in visceral gonadal white adipose tissue revealed significant increases in mitochondrial biogenesis and function terms in antibody-treated mice. In antibody-treated mice, Healthspan, a newly identified mammal-specific product promoting white adipose tissue triglyceride metabolism with S100B, was strongly upregulated. Pro-inflammatory gene expression was higher in visceral gonadal white adipose tissue of immunoglobulin G-treated mice compared with antibody-treated mice, mirroring findings in livers of IL-11 receptor knockout mice and visceral gonadal white adipose tissue of IL-11 knockout mice. Further analysis of young and old IL-11 receptor knockout and wild-type mice confirmed age-dependent pro-inflammatory gene expression in visceral gonadal white adipose tissue of wild-type mice, reduced in IL-11 receptor knockout mice across sexes. Inhibition of IL-11 extends lifespan. In parallel to Healthspan experiments, lifespan studies were conducted in male and female IL-11 knockout mice and wild-type controls until death or euthanasia, when moribund. Pooled analysis showed that IL-11 knockout mice had significantly longer lifespans than wild-type controls. Sex-specific analyses revealed significant lifespan extension in female and male IL-11 knockout mice ICA. To explore a more translational approach, 75-week-old male and female mice received monthly injections of anti-IL-11 or immunoglobulin G until death. Pooled analysis showed significantly longer lifespans for mice receiving anti-IL-11. Sex-specific analyses showed significant lifespan extension in females and males receiving anti-IL-11. Cancers are common in old mice, and gross autopsy data revealed fewer macroscopic tumors in IL-11 knockout or anti-11 treated mice. Discussion. IL-11 is upregulated across tissues with age, likely as an alarmin-type response to age-related pathogenic factors. The benefits seen with IL-11 inhibition reflect its modulation of multiple aging pathways. Although IL-11 has not been extensively studied for aging, NCPs at the IL-11 locus are associated with osteoarthritis and menopause, and IL-11 is linked with senescence and common diseases in older people. The metabolic effects of IL-11 inhibition in old mice mimic those of young mice with white adipose tissue-specific deletion of raptor. Although thermoneutrality was not studied, it is suggested that IL-11 inhibition prevents mTORC1 activation in fat, promoting age-repressed white adipose tissue beiging, while food intake and energy expenditure were excluded, and white adipose tissue beiging was shown. The specific physiology leading to weight loss with IL-11 inhibition was not pinpointed. Beyond metabolism, IL-11 inhibition improved aging features common among vertebrates, showing generic anti-aging benefits at the organismal level. Some benefits of germline IL-11 receptor, or IL-11 deletion, particularly in muscle and fat, were apparent even in young mice, suggesting primary metabolic benefits. Although cell-type specificity was not discerned, tissue-localized IL-11 activity is likely important due to its autocrine and paracrine activities. IL-11 inhibition increased lifespan in both male and female mice. The full extent of lifespan extension remains to be determined, but current data suggest that late-life anti-11 therapy increases median lifespan by over 20% in both sexes. Anti-11 was administered from 75 weeks of age, approximately 55 years in humans, and it remains to be seen if administration to older mice has similar effects, or if short-term anti-11 therapy is effective for lifespan extension, as seen with rapamycin. Old age mortality in mice is often cancer-related, and end-of-life autopsy data support the notion that IL-11 inhibition significantly reduces age-related cancers. IL-11 is important for tumorigenesis and tumor immune evasion, and clinical trials of anti-IL-11 combined with immunotherapy to treat cancer are planned. Chronic sterile inflammation is a hallmark of aging, linked with senescence and involved in age-related frailty, metabolic dysfunction, and multi-morbidity. Invertebrate studies have shown that innate immune signaling is significant in aging. Results of the study. Genetic or pharmacologic inhibition of IL-11 extends life expectancy of male and female mice. Kaplan-Meier survival curves show the cumulative survival probabilities for male and female wild-type and IL-11 knockout mice. Pooled analysis reveals that IL-11 knockout mice have significantly longer lifespans than wild-type controls. Sex-specific analyses indicate significant lifespan extension in both female and male IL-11 knockout mice. To progress to a translationally relevant approach, The effects of late-life IL-11 inhibition were examined using 75-week-old male and female mice, which received monthly injections of anti-IL-11 or immunoglobulin G until death. Pooled analysis showed that mice receiving anti-IL-11 had significantly longer lifespans compared to immunoglobulin G-treated mice. Sex-specific analyses indicated a significant extension of lifespan in both female and male mice receiving anti-IL-11. Cancers are a common cause of death in old mice, and gross autopsy data revealed fewer macroscopic tumors in IL-11 knockout mice and those on anti-IL-11 therapy. Discussion: the progressive upregulation of IL-11 across tissues with age is likely a response to various age-related pathogenic factors. The multiple benefits observed with IL-11 inhibition suggest it modulates several aging pathways, similar to the effects of polypharmacy in flies. Although IL-11 was not previously considered important for aging, genetic studies have linked IL-11 to conditions like osteoarthritis and menopause, and it is recognized for its role in senescence and age-related diseases. The metabolic effects of IL-11 inhibition in old mice are similar to those in young mice with white adipose tissue-specific deletion of raptor. While this study did not examine thermoneutrality, it is believed that IL-11 inhibition prevents MENRC1 activation in fat, promoting the beiging of white adipose tissue, which is more prominent in mice. Although specific physiology leading to weight loss with IL-11 inhibition was not pinpointed, white adipose tissue beiging was observed across genetic and therapeutic models. Beyond metabolic benefits, IL-11 inhibition improved several deterministic features of aging common among vertebrates, including frailty and sarcopenia. These generic anti-aging benefits at the organismal level suggest metabolic advantages may be primary, while cell type specificity was not determined. Tissue-localized IL-11 activity is likely important due to its known autocrine and paracrine actions. IL-11 inhibition increased lifespan in both male and female mice. The full extent of lifespan extension is yet to be determined, but current data suggest that late-life anti-IL-11 therapy increases median lifespan by more than 20% in both sexes. In these experiments, anti-IL-11 was administered from 75 weeks of age, approximately 55 years in human terms. It remains to be seen if administration to older mice has similar effects or if short-term anti-IL-11 therapy can extend lifespan. As seen with rapamycin, cancer-related mortality in old mice supports the notion that IL-11 inhibition significantly reduces age-related cancers. IL-11's role in tumorigenesis and tumor immune evasion is important, and clinical trials of anti-IL-11 in combination with immunotherapy to treat cancer are planned. Chronic sterile inflammation is a hallmark of aging, closely linked with senescence and implicated in age-related frailty, metabolic dysfunction, and multi-morbidity. Invertebrate studies show the importance of innate immune signaling in aging, supporting the potential of IL-11 inhibition as a therapeutic strategy for aging-related pathologies. Conclusions: the inhibition of IL-11 signaling through genetic deletion or pharmacologic intervention extends both healthspan and lifespan in mice. This study suggests that anti-IL-11 therapy could provide a translational opportunity to address aging pathologies in older humans. Further research is needed to explore the specific physiological mechanisms and potential clinical applications of IL-11 inhibition in aging and related diseases.